Hello and welcome to lecture number 9 of the Cardiovascular section of the Integrative Systems Physiology course. Today we'll be talking about the regulation of vascular smooth muscle tone and the implications that has for vasoconstriction, vasodilation, and regulation of local blood flow. So up until this point, we've really focused on the discussion of the cardiovascular system in the context of the heart pumping blood out to the vascular system, broad concepts of oxygen delivery and nutrient exchange, and then venous return. Now, today, what we'll be focusing on is actually the beginnings and the sort of foundations of how we direct blood flow where it needs to go. And so that is done through changes in vascular tone and vessel diameter, predominantly at the site of the arterioles. That's the major resistance point in the circulation. And so in blood vessels, we most commonly refer to this as vasoconstriction or vasodilation. So when we're looking at changes in vessel diameter and how they alter resistance in the cardiovascular system, certainly the vascular system that's dictated by Poissier's law. And so essentially what this states is that the change in resistance is inversely proportional to the change in the radius to the fourth power. So a relatively small reduction in vessel radius can result in a relatively large increase in vascular resistance and subsequently significant reduction in vascular blood flow to that region. And so that's shown here, where with the P being the pressure gradient across a given vascular bed or across a given vascular segment, R being radius, N being blood viscosity, and L being the tube length. And so critically, when you look at the things that we can actually change intentionally or inadvertently, those would be the pressure gradient is certainly one that can be readily altered through either increasing contractility predominantly or by changing the overall resistance and thereby increasing the pressure gradient to a given overall bed. Or by altering the radius. And of course, that's the major mechanism whereby the autonomic nervous system and local mechanisms alter the resistance into a given vascular region. And finally, as we discussed the other day with blood viscosity, changes in blood viscosity can have a significant impact on the amount of work that the heart has to do. And so in situations of, for example, severe dehydration or blood doping, in those instances, blood viscosity can increase significantly, and that can subsequently result in massive increases in the work that needs to be performed at a given degree of blood flow or metabolic demand. Now, similarly, when we start to reduce vessel diameter, and certainly as we start to increase blood flow, that puts us at risk of having the emergence of turbulent blood flow patterns. And certainly that generally doesn't happen for very long periods of time in normal conditions. But certainly in the case of cardiovascular disease, where you can have significant reductions in the diameter or the lumen size of the vessel with atherosclerotic plaque, or in the case of aortic valve stenosis, where the valve area significantly becomes reduced due to the stenotic valve, you can have an increase in turbulent flow when the Reynolds number exceeds a critical number. And so here, you can see here that the Reynolds number is dictated by the mean blood velocity, by the diameter of the vessel, the blood density, and then the vessel diameter. So again, the turbulence increases the pressure required for a given level of blood flow, which is important. So then instead of just having laminar flow here, where essentially the amount of work required or pressure required to drive flow into a vascular bed is relatively linear, you can see here that when we have turbulent flow patterns emerge, then we require disproportionate increases in perfusion pressure to maintain the comparable level of flow. And in addition to that, turbulent flow also does not effectively induce protective mechanisms that are induced by laminar flow in blood vessels. So as we'll discuss later, increases in the laminar flow can often increase elaboration of protective molecules from the endothelium, such as nitric oxide. And so when those aren't being elaborated because of turbulent flow patterns, that can accelerate progression of atherosclerotic vascular disease. So when we start to look at this in a bit more detail, so how is blood actually directed where it needs to go via vasoconstriction and vasodilation? We'll start with just looking at the sort of breakdown of vascular structure in a bit more detail. So with the adventitial layer, you can see that, of course, as we discussed previously, that's predominantly made of comprised of connective tissue and fibroblasts. The medial layer is predominantly vascular smooth muscle, and then the endothelial layer is comprised exclusively of endothelial cells. Okay, and when we look at vascular smooth muscle a little bit more detail, the structure of the vascular smooth muscle is and the contractile filaments within the vascular smooth muscle is radically different than what we see in striated muscle. So here you can see that actin filaments actually radiate from dense bodies. And those dense bodies are anchor points for actin filaments. And so those are analogous to Z discs in striated muscle. And so what you can see here is we've got these dense bodies here. We've got the actin filaments radiating from those. And then so between those, the myosin myofilaments are interspersed throughout the cell. So what that enables you to do now is have contraction and shortening of the vascular smooth muscle cell. But of course, it doesn't occur in the same manner or direct shortening that occurs with striated muscle. It allows for chronic increases in tension much more effectively, which thereby translates into changes in vascular tone. So again, these are not striated muscle. So now when we're looking at regulation of smooth muscle contraction, it is a bit different than what occurs in striated muscle or in cardiomyocytes. So with smooth muscle contraction, what we have is a muscle that does not contain troponin, and instead of having troponin as a primary regulatory molecule in striated muscle, now we have a molecule called calmodulin, and that is in place of troponin. And so calmodulin is a critical molecule for activating myosin cross bridges. And so calcium, what happens is calcium is a calcium-sensitive molecule, so calcium binds to calmodulin, and this calcium-calmodulin complex subsequently activates the myosin light chain kinase. And with the activation of myosin light chain kinase, that phosphorylates a regulatory chain of the myosin head, and that phosphorylated regulatory chain can now repetitively bind with the actin filament and power stroke, much like what we observed in skeletal muscle. Now, if we look at this schematically, you can see again what we have is calcium coming into the cell that can induce calcium-induced calcium release, very similar to what we observed in both cardiomyocytes as well as in skeletal muscle. That results in the formation of this calcium-calmodulin complex. That results in the activation of myosin light chain kinase. That subsequently phosphorylates myosin light chains and results in contraction. Now, to turn this cycle of events off, of course, what you would do is you would reduce calcium influx into the cell, and then this phosphatase would subsequently inactivate myosin light chain kinase and shut this pathway off, thereby allowing for relaxation. Now, that myosin phosphatase is activated when calcium ions fall below a critical level, and so, to my knowledge, there's not really a direct way to activate that myosin phosphatase. It's a calcium-sensitive enzyme, and then that removes that phosphate from a regulatory chain, and then subsequently have the cessation of cycling. So now, okay, so hopefully you should know this very well. Understand some of the fundamental differences southward from this point. And again, when we're looking at calcium-induced calcium release, it's acknowledged to be a mechanism and exists within vascular smooth muscle, but it's the critical dependence of vascular smooth muscle contraction on calcium-induced calcium release is a little bit shades of gray, because there are reports where there's dissociation of calcium-induced calcium release of that pathway in vascular smooth muscle. I would say for the purposes of the course, I would say that it predominantly focus on induced calcium release as an existing mechanism, and certainly extracellular calcium coming into the cell also resulting in activation of calcium-induced calcium release, and just acknowledging that there is some contention about the magnitude of this effect as well as how well it synchronizes in vascular smooth muscle as compared to striated muscles. So when we're looking at regional control of vascular tone, so now we have both short-term and long-term mechanisms. When we're looking at short-term mechanisms, there's one of the most commonly studied is nitric oxide-mediated vasodilation. That is a shear stress-sensitive mechanism for the release of nitric oxide. So there, as stress increases, there's an increase in nitric oxide production that results in vascular relaxation. It is also sensitive to changes in muscarinic receptor activation. So you can provide exogenous acetylcholine, and that can result in significant release of nitric oxide and subsequent vascular relaxation. And we frequently do that in my laboratory. Now, when we're also looking at short-term regulation of mechanisms that contribute to vascular increases in vascular tone and vasoconstriction, there we predominantly have adrenergic receptor-mediated vasoconstriction, and that is mediated by sympathetic activation. So that fight or flight mechanism where you end up getting increases in sympathetic nervous system outflow, that results in vasoconstriction in relatively inactive beds and the preferential redistribution of blood flow to metabolically active regions. So when we look at this from a molecular perspective, when we're looking at nitric oxide-mediated relaxation, of course, we've got nitric oxide, which binds to soluble guanylate cyclase. So that is its receptor, if you will. And so nitric oxide, when it binds to soluble guanylate cyclase and increases its activity, that increases cyclic GMP. That increases protein kinase G activation, and that subsequently results in the increased phosphorylation of myosin light chain kinase, which reduces its activity here. So you can see, as depicted down here, the phosphorylation of this enzyme at specific sites results in its inactivation. That subsequently reduces the phosphorylation of myosin light chains, and then results in, since this is inactive, then what you have is a relaxation. Also, this results in a reduction in cytosolic calcium levels by increasing the circuit 2 activation of circuit 2, which thereby results in increased storage of calcium in the sarcoplasmic reticulum. So overall, sort of a 1-2 punch of having a reduction in cytosolic calcium and inactivation of the myosin light chain kinase. And I think that's where I really want you to see, just having an appreciation, is not necessarily on these upstream mechanisms of what nitric oxide is binding to, but it's downstream mechanisms whereby it's interfering with core conserved mechanisms contributing to increases in vascular tone. Now, when we look at alpha-adrenergic receptor activation, we're predominantly looking at alpha-1 receptors when we're looking at vasoconstriction. So there, what happens is you have an increase in agonist levels, largely due to increases in sympathetic nervous system outflow. That results in increases in alpha-1 receptor activation and binding. And now what I really want you to appreciate here is that there are increases in G protein receptor coupled, so the alpha adrenergic receptors are G protein receptor coupled receptors. That increases phospholipase C. That then increases IP3. And then IP3 binds its receptor in the SR and increases calcium release, and thereby increases cytosolic calcium, and that's really depicted here. And so again, I think when we're looking at the sort of critical series of steps, you've got the alpha adrenergic receptor being a G protein coupled receptor binding, alpha adrenergic receptor binding here, increasing PLC, increasing IP3, increasing calcium release. It's a very conserved mechanism, as you'll see with other vasoactive mechanisms later on here. And of course, once we get that increase in calcium release from the SR, what we end up having is the activation of calmodulin kinase, and again, convergence of the signaling on the side of contraction. Okay, so that's how alpha-1 adrenergic receptor signaling predominantly works with increases in sympathetic nervous system outflow. When we start to look at more long-term regulation of vascular tone, what we have is the renin-angiotensin system. And so with the renin-angiotensin system, as we discussed the other day, that's a major mechanism that contributes to fluid retention and increases in vascular tone, often activated physiologically in response to significant blood flow, significant blood volume loss or plasma volume loss. So this enables you to retain blood volume and increase blood pressure through changes in vascular tone. Pathophysiologically, this is really commonly activated and hyperactivated in heart failure as a compensatory mechanism to try and increase preload. So what ends up happening is we have angiotensinogen being released from the liver. That ends up being converted to angiotensin 1 by renin, which is released by the kidney. Angiotensin 1 is converted to angiotensin 2 through angiotensin converting enzyme, which is very rich in the endothelium of the lung and kidneys. And when you have increases in angiotensin 2, angiotensin 2 is really the most biologically active molecule of the signaling cascade in terms of vasculature. So what that does is it does a number of things. One is it increases sympathetic nervous system outflow. So you have an indirect effect, which subsequently amplifies its vasoactive effects through increasing sympathetic nervous system outflow. Second, that results in fluid resorption in the kidney. Third, it results in a secretion of aldosterone, which subsequently amplifies this direct effect on the kidney. And then we also have, of course, fourth, we've got a major effect on arteriolar vasoconstriction. So there's a direct effect in the vasculature, which then increases vascular tone and thereby increases pressure. So you can think of this really as just sort of being a one-stop shop for increasing blood volume and retention of fluid, as well as increasing vascular tone, ultimately with a goal of increasing overall pressure, right? But this is a very sluggish system, if you will, and it's really focused predominantly on long-term regulation of vascular tone. So now when we look at the regional effects of renin-angiotensin system activation on vascular smooth muscle, we have increases in angiotensin 2. Those bind to the AT1 receptor, which again is a G protein coupled receptor. And then all of a sudden you should start to see things that are reminiscent of what we just spoke about before. Increases in phospholipase C, increases in IP3 binding to its receptor in the sarcoplasmic reticulum, an increase in calcium release from the SR, and ultimately increases in cytoplasmic calcium, which then activate calcium-calmodulin kinase and then increase this shift towards active myosin light chain kinase and vascular contraction and increases in vascular tone. Now, importantly, with the renin-angiotensin system, there are also balancing mechanisms there that act locally as well. So the AT2 receptor, over here we're talking about AT1 receptor activation, which is deleterious in the context of disease. The AT2 receptor is actually beneficial in many ways. And so there, what we have is an increase in angiotensin, the AT2 receptor activation. Again, it activates via a G protein coupled receptor, but now what that does is preferentially increases nitric oxide synthase activity, increases NO release, increases bradykinin, and ultimately results in increases in vascular relaxation. And so there are efforts to develop AT2 receptor selective activators, right? and avoid the activation of the AT1 receptor. And similarly, there are AT1 receptor blockers that are used clinically that enable the blockade of AT1 receptors, but also the activation of AT2 receptors via endogenous angiotensin 2. So again, if you have these increases in nitric oxide, then you'll end up having the increases in the soluble guanylate cyclase, increases in CGMP, increases in protein kinase G, and then the inactivation of myosin light chain kinase. So in addition to these sort of ligand-driven and molecule-driven changes in vascular tone, there are also mechanisms that contribute to regulation of blood flow through autoregulation of local blood flow in response to changes in pressure. So there, what we see are independent of neural and endocrine signaling mechanisms. So Here you can see that what you have with autoregulation is you have increases in arterial pressure, and at very low pressures you have a relatively we'll say linear dose response mechanism, if you will, between changes in arterial pressure and blood flow. So here, increases in pressure, increased blood flow. You end up getting to a point at which you have somewhat of a plateau here, and then all of a sudden you have this relatively large autoregulatory range where you can have very significant changes in blood pressure that don't result in changes in blood flow. And so this is really the autoregulatory range, predominantly a physiological range of blood pressures. And so if you exceed this, what that does is then, of course, you'll start to have this uptick in pressures here again. But when we're looking at regions like the brain circulation, for example, what you don't want to have is this totally linear relationship between blood pressure and blood flow, because certainly there, should you have an acute stress response and an increase in pressure, you want to be able to maintain and match blood flow supply and nutrient supply to the brain. And not overperfuse the brain, since that would result in profound changes in pH in the brain. And so there, it's got a relatively wide autoregulatory range across which either increases or reductions in flow have a relatively negligible impact on blood flow. So again, as I just mentioned, the two major areas in which this is particularly important is both in the brain and in the lung. Right, so in the brain, you want to be able to not over or under perfuse. In the lung, you want to still be able to direct blood flow to regions that are well oxygenated and ensure that gas exchange occurs efficiently. So now, finally, when we're looking at skeletal muscle autoregulation in terms of autoregulated beds, there, when muscle contracts, you have generation of metabolites. That reduces the partial pressure of oxygen due to increases in oxygen consumption, and it results in very rapid vasodilation and to increase nutrients and oxygen delivery to those beds. Further, the return of basal levels of flow is via an autoregulatory mechanism as well. So there, again, when we're looking at local matching of blood flow to metabolic demand, that's an autoregulatory mechanism, primarily driven by PO2 and metabolite levels. So when we're looking at brain autoregulation, it's a bit different. So of course, when you think, well, what's the brain vasculature sensitive to? It's really sensitive to changes in partial pressure of oxygen and the partial pressure of carbon dioxide, which is critical for acid-base balance. And this is being driven by the Henderson-Hasselbalch equation. So what you can see here, again, as you increase CO2, you're going to be increasing hydrogen ion concentration and vice versa. And so for the brain, it's absolutely pivotal to make sure that you're appropriately maintaining blood flow matching to metabolic demand, so that way you don't have profound changes in brain pH. So what you can see here is as you decrease CO2, you're going to be increasing brain pH, or having more basic brain pH, unless blood flow is reduced in proportion. And so that's why when individuals hyperventilate, you can oftentimes feel lightheaded because you're blowing off a bunch of CO2, and subsequently the brain is going to vasoconstrict, and it's a profoundly powerful mechanism to reduce brain blood flow, and does so even in the face of reductions in local PO2. The pulmonary vasculature, when we're looking at pulmonary autoregulation, again, this is an area where the regulation of blood flow is precisely the opposite of what you normally would see. So here, with a decrease in PO2, you have vasoconstriction, because what you want is blood flow going to regions of the lung that are very well oxygenated. And so there, where you have a reduction in PO2 in the alveoli, you don't want blood going there, so you have vasoconstriction. And that's really the only area in the body where this relationship occurs. And so it's, again, exactly the opposite of what you anticipate seeing elsewhere. So when we're looking at key cardiovascular variables that are regulated, of course, you think, well, is it blood flow? Is it blood volume? Is it blood pressure? Is it PO2? Is it PCO2? Importantly, we really don't have measures. We don't have regulators of blood flow per se, or blood volume per se. What we have is blood pressure or distension of certain regions of the body, and then subsequent changes in PO2 or PCO2 at the chemoreceptors. And so we've got baroreceptors or pressure receptors and chemoreceptors that are critical to the maintenance of cardiovascular homeostasis, and that's what we will start to move towards in subsequent lectures, discussing in more detail. All right. Well, thank you again for your attention to this, and I will look forward to seeing everybody soon. Thank you.